Okay, welcome everyone. So this is a follow-up from last week's presentation by Prof. Teresa Rousseau. This is one of her students. Louise is also a supervisor of Crystal in the audience. She did some metabolomics work on HIV, and she's going to tell us more about her master's project. But just to give a short introduction on her, uh, where did I open your? Okay. I lost your profile. How did I do that?
I can probably introduce myself if you would like me to.
I found it. So
Okay.
Crystal Roux, so she attained her MSc last year, 2022, in Medical Immunology. The study focused on the comparative effects of antiretroviral treatments, including some drugs there. I'm not going to pronounce them. There's three there. Using metabolomics, cytokine profiles, and platelet activation in people living with HIV. So the technique she used, so untargeted NMR, which is close to my heart, bead-based suspension arrays, and enzyme-linked immunosorbent assays, which are more commonly known as ELISAs. So some long words there, complex words. And I think you can start sharing your screen, Crystal, your side.
Good morning. Can everybody see my screen?
Yes. So I'm going to hand it over to you, Crystal, and you can tell us all about your master's study.
Great. Thank you so much. And thank you for giving me this opportunity to share. Today I'll be discussing the comparative effects of two antiretroviral regimens, namely Efavirenz and Dolutegra, on metabolomics, cytokine profiles, and platelet activation. So this is a little bit of a subset of what we did in my master's. So yes. So as we all know, HIV is a large problem, particularly within the South African population. According to recent 2021 Joint United Nations Program on HIV/AIDS statistics, approximately 37.7 million people are living with HIV worldwide, with 7.8 million being accounted for in the South African population. The introduction of combination antiretroviral treatments led to a decreased number of AIDS-related deaths, with approximately 650,000 AIDS-related deaths in South Africa within 2021. However, mortality rates associated with non-AIDS-related diseases have increased in people living with HIV. Studies have reported that non-AIDS-related deaths account for approximately 53% in high-income countries, 34% in developing countries, and 18.5% in sub-Saharan countries. Of these, the majority of deaths are related to cardiovascular disease, non-AIDS malignancies, and liver disease. So in 2019, the World Health Organization recommended the transition from Efavirenz-based antiretroviral treatments to those containing Dolutegra. Efavirenz is a non-nucleoside reverse transcriptase inhibitor, which directly inhibits HIV reverse transcriptase through binding to the enzyme. In contrast, Dolutegra, an integrase strand transfer inhibitor, inhibits integrase's ability to insert viral DNA into the host's DNA. The transition was recommended due to the reported superiority of Dolutegra-based regimens regarding increased viral suppression rates, low drug-drug interactions, reduced side effects, and high barrier to drug resistance compared to efavirenz-based treatments. Unfortunately, concerns have emerged on possible side effects of Dolutegra, especially pertaining to weight and cardiovascular disease. So therefore, for our study, we aim to investigate the effect of the two treatments on the metabolites using untargeted nuclear magnetic resonance. We also have the cytokine analysis for inflammation, as well as the platelet activation with the ELISAs. So we collected whole blood samples in EDTA tubes, which were centrifuged. Plasma was then stored at minus 80 degrees Celsius until the use. We obtained informed consent from all participants, and the study was approved by the Faculty of Health Science Research Ethics Committee at the University of Pretoria. So 5 people living with HIV attending Tshwane District Hospital between 2020 and 2022 were included in the study. The participants were over 18 years of age, virally suppressed and receiving Efavirenz-based treatments comprising of tenofovir, disoproxil fumarate, and emtricitabine in addition to Efavirenz. These participants transitioned to the dolutegravir-based regimen composed of tenofovir, disoproxil fumarate, lamivudine, as well as dolutegravir. Samples were collected 6 months after the transition to investigate any changes. 16 healthy HIV uninfected student volunteers and staff at the University of Pretoria were used as controls, and the exclusion criteria included elevated C-reactive proteins and CD4 T cell counts of less than 150 cells per microliter. Of note, there were no significant differences between any of these groups, except for the slight imbalance within the gender, but this correlates with the current statistics in South Africa, in which females have a greater incidence of HIV. We did paired and unpaired non-parametric tests using RStudio with a 5% significance level. We did paired comparisons between the Efavirenz treated cohorts at baseline and 6 months thereafter. In addition, we also we performed paired analysis between the Efavirenz treated and dolutegravir treated cohorts, and we did unpaired analysis between the controls, the Efavirenz treated, and then the controls and the dolutegravir treated participants. So firstly, we did metabolomics with untargeted nuclear magnetic resonance. In short, nuclear magnetic resonance is based on a magnetic dipole similar to magnets with a north and south pole generated by nucleic charges. As a strong magnetic field is applied, the nuclei are randomly orientated, causing the dipoles to face one direction. The energy loss after applying the magnetic field is recorded as oscillations and depend on the nucleus being detected, the magnetic field strength, and the chemical group and electron shielding surrounded the nuclear element. The data obtained as chemical shifts can then be analyzed and compared with various softwares and metabolomic databases. So what did we do? The method used was in accordance with the standard operating procedures of the Center of Human Metabolomics, with the methodology adapted from Mason et al. Prior to sample preparation, the centrifugal filters were rinsed 5 times with distilled water. Thereafter, 1 milliliter of samples were added to the centrifugal units and centrifuged. A 2-fold dilution of the filtered sample solution was made by adding 300 microliters filtered sample to 300 microliters buffer solution. The mixture was then vortexed and centrifuged. Finally, 540 microliters of the ultrafiltrate was transferred to the 5 millimeter NMR tube, which was sealed and loaded into a sample express autosampler in a randomized order with the quality controls interdispersed. Samples were measured at 500 megahertz on a Bruker Advanced 3 HD-NMR spectrometer. The spectrometer contained XYZ gradient coils, as well as a 5 millimeter triple resonance inverse probe head able to detect hydrogen, nitrogen, and carbon atoms. The inner coil of the probe was optimized for hydrogen observation, which was our focus. Hydrogen spectra were acquired after 128 scans with 4 dummy scans. Additional settings included acquisition time of 2.72 seconds and receiver gain of 64 seconds. The sample temperature was maintained at 27 degrees Celsius approximately, and the water resonance was pre-saturated by single frequency irradiation. The results obtained were represented on an NMR spectrum with the vertical axis representing the intensity of the compound and the horizontal axis indicating the chemical shift recorded in parts per million. Bruker Topspin and Bruker Amex software were used in the analysis to analyze the data, and annotations and analysis were further completed with MetaBioAnalyst. Once factor statistical analysis was performed on MetaBioAnalyst, a total of 417 spectral bins were identified, after which 295 features were excluded. These are those that fell 20% out of the relative standard deviation of the quality control samples. Thereafter, the data was log transformed and autoscaled, and ANOVA was then performed, resulting in 16 bins of interest. Paired and unpaired analysis were performed on the bins of interest, and yes, so our results then indicated through paired analysis, we identified one metabolite of interest, namely 3-hydroxybutyrate, which showed a 2-fold increase after 6 months of dolutegravir-based treatment. Of note, a larger difference was observed between the new treatment regimen and the control group. So briefly, 3-hydroxybutyrate is a ketone prevalent as an energy source in the absence of glucose. It also functions to regulate energy expenditure during malnutrition and is responsible for signaling through its effects on DNA methylation. The elevated levels observed in the dolutegravir-treated cohort may have had a large range of effects, both positive and negative. Specifically, it can result in diabetic ketoacidosis, hypertension, and cardiovascular disease improvements, neurological disease prevention, as well as a promotion or inhibition of cancer, depending on what type of cancer it is. The following table is a summary of the unpaired analysis between the 2 treatment groups and the control group. The arrows indicate either an increase or decrease in the detected analyte, and a single arrow, yes, yeah. The single arrows indicate a fold change of less than 1.5, whereas 2 arrows indicate a fold change of larger than 1.5. So as observed, the Efavirenz treated cohort had altered levels in adenosine monophosphate, creatinine, formate, glucose, L-lactate, urea, and 1,7-dimethylxanthine compared to the controls. In contrast, the dolutegravir-treated group only had significantly altered levels between formate, urea, and 1,7-dimethylxanthine when compared to the control group. 1,7-dimethylxanthine is a metabolite associated with caffeine metabolism, possibly due to coffee intake prior to blood collection. Then we looked at the results, the discussion. So these differences indicate a potential effect on energy metabolism, which involve the D-glucose, adenosine monophosphate, lactate, formate, and 3-hydroxybutyrate levels, as well as kidney functions, which will be discussed on the next slide. Specifically, the elevated levels of D-glucose and adenosine monophosphate in the Efavirenz-treated cohort may be as a result of the drug's mechanism of action, which inhibits cellular D-glucose uptake and complex 1 of the electron transport chain pathway. As we observed, the transition to dolutegravir-based regimens appear to normalize both the D-glucose and AMP levels to those observed in the control group. This is potentially due to the alternative drug mechanism utilized by dolutegravir, in which it chelates magnesium. So reduced levels of intracellular D-glucose levels observed in the Efavirenz-treated cohort may result in cellular energy generation through ketogenesis. This is indicative of the elevated formate levels. The elevation of 3-hydroxybutyrate after the transition to the dolutegravir-based regimen may be as a result of sufficient intracellular D-glucose levels for energy production, meaning that 3-hydroxybutyrate is no longer required at such high quantities for ketosis. Interestingly, an increase in ketosis has also been found to decrease muscle glycolysis and L-lactate concentrations, which could explain the reduced levels of L-lactate observed in the Efavirenz treated cohorts. So in addition to the energy metabolism, the metabolites indicated changes within the renal health. Specifically, chronic renal failure is a common complication associated with HIV infection, with it affecting between 3.5 and 48% of people living with HIV. So creatine levels are often used to indicate kidney function and health, as it is excreted as waste following the breakdown of creatine in muscles and cells. Therefore, based on the normalization of creatinine following the transition, it would appear that dolutegravir is beneficial to kidney functions and indicates an improvement from that observed within the Efavirenz treated cohorts. Importantly, creatinine and urea levels can be influenced by many factors, including diet, liver function, skeletal muscle mass, disease severity, and renal blood flow. And as such, the interpretation of these results are speculative. Secondly, we looked at the bead-based suspension array for the detection of cytokines and their quantification. So this assay allows for the simultaneous detection and quantification of up to 100 analytes. The assay uses distinct beads with known probe molecules on their surface to which analytes bind. These analytes are detected and quantified using fluorophores, which fluoresce and can be detected and analyzed with software. So the study made use of a bead-based BioPlex human cytokine assay kit with a methodology in accordance to the manufacturer's instruction manual. In summary, plasma samples were thawed and diluted fourfold with sample diluent. Thereafter, the magnetic beads were added to each of the 96 wells. The plate was washed using an automatic magnetic microplate washer. Thereafter, samples, blanks, standards, and controls were added to the designated wells, and the plate was incubated on an orbital plate shaker and then once again washed. The detection antibody was then added to the well. The plate was incubated and washed once again, and streptavidin and phycoerythrin was added to each well and incubated. Lastly, the plate was again washed and the beads were resuspended in order to be analyzed. The plate was then inserted into the Biorad Luminex 200 suspension array system, which has two lasers that excite the fluorophores. Specifically, the red laser detects the presence of the analyte through the bleed fluorescence, whereas the green laser excites the detection antibody, which quantifies the analyte. BioPlex Manager Software 6 then determined the concentration of each analyte. So it was determined that through paired analysis that there was no alterations of significance before and after transitioning. However, unpaired analysis depicted differences in two cytokines when compared to the control group. These are the granulocyte monocyte colony stimulating factor and then the platelet-derived growth factors. So, which is now depicted here. Following the transition, we observed that the factors decreased. However, they approached levels that were similar to that observed in the control group. So both granulocyte macrophage colony stimulating factor and platelet-derived growth factors are involved in cell proliferation and differentiation. Elevated levels of GM-CSF, which is the abbreviated form of granulocyte macrophage colony stimulating factor, are commonly associated with chronic inflammatory diseases such as arthritis, osteoarthritis, inflammatory bowel disease, and multiple sclerosis. Interestingly, GM-CSF interacts with adipose tissue macrophages, closely related to metabolic dysfunction, impairment of adipocyte function, and insulin resistance, which can also result in type 2 diabetes mellitus. GM-CSF also functions in cholesterol clearance. Then we have our platelet-derived growth factor. which is a cytokine that is involved in tissue repair and regeneration following an injury. Studies have identified a positive correlation between the platelet-derived growth factor concentration and coronary artery disease severity, platelet rupture incidence, platelet activation, and thrombotic events. Lastly, we did a sandwich enzyme-linked immunoassay, abbreviated ELISA, and we investigated the quantity of soluble cluster of differentiation 40 ligand and regulated on activation normal T cell expressed and secreted, which is abbreviated RANTES. So ELISAs are commonly used in detecting and quantifying hormones, cytokines, antibodies, and antigens. The assay uses a 96-well plate, which is pre-coated with antibodies to which the antigen binds. Subsequently, a detection antibody is added, which binds to the antigen and allows for the binding of quantifiable enzymatic labels detected at a specific wavelengths. So this current study made use of two ELISAs, namely the Ray Biohuman CD40 ligand ELISA kit and eLab Science human RANTES ELISA kit. And once again, the assay were conducted in accordance with the manufacturer's instructions. So prior to starting the assay, the plasma samples were thawed and diluted 3-fold and 20-fold for soluble CD40 ligand and RANTES, respectively. Sample standards and blanks were added to the designated wells. The plate was incubated with gentle agitation and each well was washed. Detection antibody was added to each well, followed by incubation and washing. Subsequently, horseradish peroxidase streptavidin solution was added and the plate was once again incubated and washed. Thereafter, the tetramethylbenzidine substrate solution was added. And then the change in color was observed. So as you can see here in the specifics pictures, this was the color change that was observed. It went from see-through to blue. Upon reaching the optimal color, we added the stop solution, which then results in the color change to yellow that you can observe there on the right. So the optical density of each sample was determined using the power wave microplate spectrometer set at 450 nanometers. A standard curve was generated. We're using GraphPad Prism software, and then the samples were compared to that in order to determine analyte concentrations. So once again, no changes were observed, while significant changes were observed during paired analysis. However, upon comparing the efavirenz treated to the control groups, there was a significant difference, which then disappeared following the transition to dolutegravir. So we have contradicting results have been reported on the effects of different antiretroviral treatments on platelet activation, with some authors implicating antiretroviral treatments in the upregulation, downregulation, or persistence thereof. However, people living with HIV, irrespective of viral suppression, normally present with chronically elevated platelet activation markers. This, which is indicative of platelet activation. This we observed within our efavirenz treated cohort. The increased platelet activation in the efavirenz treated group is reportedly associated with the mechanisms employed by the specific treatment regimen in which it directly affects platelet activation. This same finding was observed by Davidson et al., who established that efavirenz-based treatments are associated with increased systemic concentrations of soluble CD40 ligand, and that dolutegravir reduces platelet activation and causes a reduction in degranulation. The decrease in platelet-derived growth factor that we also previously observed with the cytokine analysis could also have influenced the platelet activation. So to conclude, was this transition a a step in the right direction, and what are the potential implications on non-AIDS-related diseases. So based on our results, we established that the metabolic profile pertained to energy metabolism and kidney function improved to levels observed in the healthy HIV uninfected cohort. The effect of increased concentrations of 3-hydroxybutyrate is still unknown, and I believe only time will tell what this effect will be. Secondly, inflammatory responses remain largely the same, with slight improvements in granulocyte and macrophage colony stimuli factors and platelet-derived growth factor concentrations. And lastly, platelet activation declines to levels observed in the control group. These improvements would likely be associated with reduced chronic inflammatory disease, metabolic dysfunction, improvement, plaque rupture incidence, platelet activations, thrombotic events, and reduced risk of developing insulin resistance. So, although this study indicates benefits associated with the transition, other factors that were not considered or studied may still result in elevated disease risks. For example, new data has indicated that dolutegravir does not result in lipid accumulation, but instead reduces adiponectin expression and secretion from adipocytes, which may develop into cardiovascular disease and type 2 diabetes mellitus. Moreover, the small sample size used in the study may result in false representation of the population, and as such, further studies can focus on larger cohorts. The detection of 1,7-dimethylxanthine has emphasized the sensitivity of NMR to an individual's dietary intake, and as such, further investigation can be aimed at determining these effects. I just want to say a large word of thanks to my supervisor, Helen Steele, as well as my co-supervisors, Jan Renal, and then also Louise, that's on the call, and then as well to Mika, who actually also helped when I was doing metabolomics to collect some samples, and a big thank you to Shane for helping with all the analysis of the NMR results and for allowing me doing the training with me. So yes, as well, I would like to thank the Northwest University for their facilities, and then my funders, which were the University of Pretoria and the NHLS. So thank you for listening. Are there any questions and comments?
Thank you so much, Crystal. It was a very good presentation, very interesting. As with all the previous journal clubs, we encourage the audience to ask questions. Either you can raise your hand, ask directly, verbalize your question to Crystal, or you can type it in the chat. I see Crystal has provided her email address there. So anyone interested in her study or follow up on her study or anything related to what she has done here, or any questions on what she's done, you are more than welcome to contact her. While we wait for questions, I have a question for you, Crystal. Have you done any correlations between the cytokines and the metabolites?
Not well. The cytokine are already, there is correlation between them because some of them are pro-inflammatory, so those ones will go together, and other ones are anti-inflammatory. So those ones we didn't do because that's mainly been done. In terms of between cytokines and metabolites, I haven't done that, no, which is I think quite a nice thing to do. Yeah, we did do some correlations in terms of weight and gender and those things, but there was no real correlation between that.
Okay. I think it'll be interesting just to see if any of those two cytokines that you identified, if they're responsible for some of these metabolites.
Yes. Yeah. Definitely. I think only thing is, I think the biggest thing is that the significance was actually very, it wasn't very clear. So I don't know if it's, yeah, I don't know. But yeah, sorry.
And what is next for Crystal, the master's graduate?
So I'm currently doing my HPCSA internship, and then I am going to start with my PhD sometime as well, where I'll be looking at comparing the HIV-exposed infants to HIV-unexposed infants in terms of T regulatory cells, moving more towards single cell work, genomics, and also remaining with cytokines as well. So yes, Louise did the metabolomics of that part.
Yes, yeah, she did a lot of work there, but I remember you also did a lot of work in our lab.
Yes, unfortunately, with all the so we started off with quite a few samples, but as we started doing analysis, we started to see that there were some problems, like some of them had elevated C-reactive proteins, which could then indicate inflammatory response due to something else and not treatments associated. So that's how we ended up with 5 at the end.
Okay. Fidel says nice work. Thank you for joining us, Fidel. I think you are in Canada, aren't you? The big time difference there must be about 5 in the morning. Thank you for joining us. So are there any other questions from the audience? If not, I also want to thank our speaker again. So I wish you all the best with your PhD research. Hope to have you again on Journal Club. You can present on that in the next 2 or 3 years.
Thank you.
After you're done with that, I know Louise is going to present soon on her PhD as well. And it'll be very interesting to see the outcome of that. So you guys are doing very interesting research, incorporating metabolomics into HIV research. So that's great.
Yes, it's exciting. But thanks again for your help and input.
No problem. Okay. So yes, contact Crystal if you want to find out more from her. And we'll put this recording up on YouTube if you want to listen to it at another point. So join us next week. We're going to have another master student presenting. That will be on a ketogenic diet in chronic alcohol-fed mice. So another health hot topic, intermittent fasting. So please join us again next week. Thanks so much, everyone. Have a good day.
Cheers.